Hi, my name is Matthew Lykins, and today I'm going to be analyzing the effects of bioluminescence on the photosynthetic process in eukaryotic cells. So, to start, as stated by the Biological Bulletin, identifications of particulate light sources in bioluminescent cells is needed to understand mechanisms and regulation of light emission. The concept of bioluminescence is a natural phenomenon that occurs in the world of biology. Some organisms contain these bioluminescent properties naturally to help them survive in the nature, such as fending off organisms, finding mates, and other abilities. One of the most common organisms that contain these bioluminescent properties is called Pyrocystis Fusiformis. Pyrocystis Fusiformis is a blue-green bioluminescent dinoflagellate, which is a type of algae, commonly found in nature and has been commonly used in research due to its bioluminescent properties. However, there is a lack of knowledge when it comes to the bioluminescent properties in Pyrocystis Fusiformis when it comes to their effects on neighboring organisms. To continue, photosynthesis is a natural phenomenon that occurs in many eukaryotic cells. In particular, Chlorella Vulgaris is a famous species of eukaryotic cell that is known for its oxygen emission and has even been considered a contender for the Institute of Space Systems, as shown by the source by Gisela Dutrell. Moreover, photosynthesis requires an activation energy to start the reaction, which has normally occurred from the use of sunlight, as can be shown in the diagram below, right here on the light. But when it comes to the effects of bioluminescence on neighboring organisms on the photosynthetic process, there is a gap in grounded research, which would lead me to my research question: can the bioluminescence from bioluminescent dinoflagellates, Pyrocystis Fusiformis, start the photosynthetic reaction in eukaryotic cells, Chlorella Vulgaris? In order to fully understand this, we first need to analyze all existing literature. First of all, as starting on the left, under chlorella growth, this source would cover the influence of light on Chlorella Vulgaris, and if Pyrocystis Fusiformis can actually start the photosynthetic reactions. This would help me to kind of understand if Chlorella Vulgaris, by its nature and the known effects of how it works, and whether light or not, or artificial light can start its photosynthetic reactions, and kind of start that presentation of a gap for me. And on the right, this source would state the nature of the bioluminescent algae and would reveal that while there was knowledge on the bioluminescent algae and how it was activated, there was lack of knowledge when it came to its effects on neighboring organisms, and especially eukaryotic cells in this case. To continue, the source on the left over symbiosis would kind of give the further questioning and inquiry over my gap, and kind of a wondering of, can Pyrocystis Fusiformis and its bioluminescence finally or be harnessed to create oxygen emission, and vice versa to create a symbiotic relationship between the two plants artificially. And on the right, a source that would be constantly used throughout this study was a photosynthetic measuring source, which would help to measure oxygen emission, photosynthetic activity, and overall the results from this research. So my hypothesis is that in cellular biology, chloroplasts start the reactions by the absorption of light, and they are usually 35% efficient in doing so. But when it comes to bioluminescent dinoflagellates, there is a small amount of intense light that is produced. So if photosynthetic reactions might be small and incremental if they happen, however, it can be inferred that a photosynthetic reaction will occur from the bioluminescence of dinoflagellates, which would be Pyrocystis Fusiformis. So my methodology was an experimental method with quantitative results. And to start, there were 4 steps that went into my method. The first would be the growth of organisms, the second being a creation of an apparatus, the third testing, and the fourth comparison between numerical data. To start, the growth of organisms. There will be 2 organisms that are used in this experiment, including Chlorella Vulgaris and Pyrocystis Fusiformis. These 2 will be grown under a maximum growth condition in order to achieve maximum readings for when the tests are performed, and the maximum growth environments will be included below, which were also used in this study. Secondly, the creation of an apparatus. This apparatus will be constructed that can measure photosynthetic activity, which was also used in the existing literature review to understand how to measure photosynthetic activity. The device is called an Otis apparatus, and it will be used to measure any oxygen that comes off the Chlorella Vulgaris for both my experimental and control group that will be shown later. And the particular AUDIS apparatus that will be used in the study has been constructed in a closed-loop configuration, which will trap all possible oxygen production inside the system and allow for all to be read there. To continue, this apparatus works by placing the sample in a small test tube, as can be seen on the right, and any oxygen that will produce will go up the blue rubber tube, which will be full of distilled water, and any water that is displaced by the oxygen will be dripped out of the right side onto a drip pan. And the amount of oxygen that is displaced will then be measured via a ruler, because it is a milliliter tube, so it will be able to be transferred from a measurement into a volumetric measurement, and it will be produced over 3 minutes, which was the recommended time by AUDIS. And then third, testing. The experiment will be sorted into 2 groups, Chlorella Vulgaris alone to create a control group, and then Chlorella Vulgaris within the presence of Pyrocystis Fusiformis, which will be our experimental group. The group of just Chlorella Vulgaris will be placed in the apparatus, and a 3-minute test will be performed to obtain a control variable. Next, the group of Chlorella Vulgaris will be placed in the AUDIS apparatus, and the Pyrocystis Fusiformis will be placed in a magnetic stir in order to keep constant agitation during the experiment for multiple testings. However, there is a lack of knowledge when it comes to the use of these magnetic stirs, and a constant agitation speed will need to be discovered before these can be used. And the Pyrocystis Fusiformis will be placed as close as possible to the Chlorella Vulgaris to maximize chloroplast absorption and possible photosynthetic activity. And then fourth and lastly, the data will be recorded in the table above and read in milliliter of displacement from the AUDIS apparatus, as was said before, which can be then used to analyze the occurrence of photosynthetic reactions in Chlorella Vulgaris under the presence of Pyrocystis Fusiformis. So, what are my results? So, from the found data, it can be seen that in the Chlorella Vulgaris by itself, there would be no oxygen produced over the 3-minute period. However, with the Chlorella Vulgaris, it wouldn't be much different, and there would be less than 0.1 milliliter of an oxygen production, and it was nearly impossible to read via the ruler. Also, it could be seen that there was approximately 20 seconds or lower of actual bioluminescence time, and that the bioluminescence that came from these species would be very hard to go over any higher that time, and intensity of light would decrease drastically, and the 3-minute period was practically impossible to get. So my results in discussion, it was found that no matter the length of the bioluminescence during the test, that agitation was not enough to start a noticeable photosynthetic reaction. This debunks my original hypothesis that rather states that bioluminescence is not able to give, or is able to give an activation energy, and instead states that it's not enough energy to actually start the photosynthetic reactions that are necessary. Given that Pyrocystis Fusiformis is unable to start the photosynthetic reactions within Chlorella Vulgaris, this means that any possible symbiotic relationships are not possible, and also will be very improbable even if other considerations are put into mind. Also, some additional findings would be that while trying to find the best speed for a magnetic stir, that 700 RPM would create the highest bioluminescence, as can be seen on the right. And also, if placed over 700 RPM, these cells would be sadly killed, as they were seen below. When compared to cells that are live, it can see that rotational force, which has yet to been found by proven and accurate study, can drastically kill and shatter these organelle walls. Some limitations, however, did come into this study. To start, purging the apparatus was very difficult, and it was hard to get all the air bubbles out of the actual system before trying to measure results and get final understanding. There was also not a set time of growth that were set, and rather, instead of setting them for a set amount of time, the organisms were looked for livelihood and would see whenever we started seeing activity and noticeability amongst whether they were glowing or whether they were growing in that environment rather than an actual set amount of time, which could have created some possible error. And also there was a lack of knowledge on magnetic stirrers, which would leave the death to a couple of cultures before the study, and bioluminescence was short, not being able to go to the 3-minute time presented by AUDIS in the beginning of the study. So future directions that come is that it can be inferred that any other color range except far red would be ineffective, and when compared to this study would not be enough to start photosynthetic reactions at a noticeable level. Despite the blue range being the most powerful when it comes to light emissions in terms of quantum energy, far-red radiation should be instead used if trying to facilitate oxygen emission inside eukaryotic cells, which can be shown in the source over quantum requirement by George Schmid below. Intensity of light of Pyrocystis Fusiformis was a maximum of 700 RPM in the magnetic stir, and when trying to achieve maximum light from these organisms, that should be taken into consideration for future study. And when it comes to symbiotic relationships, bioluminescence from these dinoflagellates cannot be used as an artificial light source for eukaryotic cells, which my expert advisor, Ms. Klingbeil, professor of biology at Lipscomb University, would help me to get through this study and give me the resources I need. And here are my sources.
All right, Matthew, thank you so much. All right, oral defense panel question number 1. Matthew, how did the choices you made when designing or implementing your research method impact your research process?
Okay, so my method was a highly experimental method, and coming with that design, there's only really one way to kind of do this, and other ways are a little bit more improbable, but with an experimental method, especially with this, it would really start to kind of affect my process, and I would start to learn halfway through that with this experimental process, that I was looking for more than just if photosynthetic reactions could start happening, but I was looking at more of how these species started behaving in their actual groups. And I started being able to find more than what I actually expected from my hypothesis and my process. And by using this experimental group, I realized that I was able to find a lot more than I initially intended for, and my process would be highly influenced by so, because even though it was very structured, there'd be a lot of different data that would come off of it that would need to be analyzed.
Hi, Matthew. What are the real-world implications or consequences related to your findings?
Okay, so in my case, a lot of this bioluminescence studies, bioluminescence is kind of a little bit understood in this in the world of biology, and there's a lack of information when it goes to how it reacts with the actual system and environment around it. And when it comes to this study, it was more than just seeing if it'll react with other organisms, but also how this species can react in its best rotational force, and also how just overall more information of the organisms, which really helps for future understanding and kind of give a scientific understanding of these new organisms.
How did your expert advisor facilitate your deeper understanding of the research process?
Okay, so I went very kind of blindly in at this at first, and my expert advisor, without her, I would have struggled a lot, because firstly, I started with fluorescent proteins to start with this, and that very, very well died out and shifted the entire process that I was going for. And throughout, she would really help me to kind of understand what I was doing, help me to organize it and realize the reality of my research, not just kind of helping me along. She would realize to help me actually understand what I was doing, and trying to help me guide on along and help me to ultimately get to my results. And she was a lot more than just material help, but rather a help for the research study and process overall.
Matthew, thank you so much. All right, guys, the floor is open for general questions.
Hey, Matthew.
Hi.
So do you think the reason you could probably only get about 20 seconds is because the little critters ran out of their...
Oh, no, it definitely is.
You know?
No, it 100% is. They run out of their, it's luciferin. That it's a chemical compound that combined with oxygen will produce the bioluminescence. And whenever they would run out of that luciferin chemical, which is produced during their day cycle, they would run through it too fast, and then the bioluminescence would go from really intense, as could kind of be seen right there, and it would die out almost to like a bluish green. And I wish that they could go for longer, but I couldn't get any longer than 20 seconds. And that is a big limitation. John. Sorry, I totally skipped you.
No, it's okay.
How did you figure out to did you slowly ramp up to 70 RPM?
Yes, okay, so with that, it was, at first, I had to find a way to where I could accurately measure them at a certain intensity of light, because all the sources I said how you can activate them, but not which way is best to activate them. And I would slowly get it up, and I would turn it as quickly as I could, frankly. I would turn it ramp it up, so it would kind of go immediately to that, because immediately once you start activating them, their energy dies out. So you have to activate them really quickly. And I realized that 700 would be it, but if you go over, they all die. It just happens. They all die. All right.
Any other questions? So I was wondering, if you could measure that wavelength, right? What that wavelength is that they're producing, and then artificially produce that.
Yeah.
Then you could potentially maybe have longer access
Mm-hmm.
than 20 seconds.
Yeah, and that is definitely true. There were some, but it was large ranges over, from the sources I found, large ranges of wavelength. And I guess this was more of an understanding of how the environment can really do it rather than the actual species understanding. And when it came to, it was really difficult, because wanting to artificially doing it is really difficult, because a lot of the sources that were provided would it would start specifying the exact amount of photons that you needed, and it was very strange for doing so. And it was a lot easier and more simplistic to be able to do it naturally.
Guys, any other questions?
Are there different colors of diamoplasma?
I wish. I really wish there were. And and if there are, they're very, very rare. And that's why I also said, with the red, there needs to be kind of an understand, because far red is known to kind of do the same thing, if I can remember where I put it. Right in there. So far red is known to kind of do the same thing. Blues and reds go together to help with growth of organisms. And reds are kind of understood to be really, really good at giving most of the energy when it actually comes to plants. But the only red bioluminescence I could find were in rare species of jellyfish. And I'm not going to get rare species of jellyfish.